thank you very much for the organizers for inviting me here. And I'm going to give a research talk. And this is a seminar, so everything is not going to be very polished. There's 1/3 of the work is published, and the other bits are either being written up at the moment or under review. So please stop me at any time if you have questions. You might have heard from different public websites and news agencies that drug resistance is becoming a huge issue. So we have now lots of antimicrobial resistance, and we don't have any more pathogens that we used to be able to treat are now resistant to treatment. And this is not only a problem for bacteria or viruses, but it's also a problem for cancer. So essentially, cancer and all infectious disease show the emergence of resistance to drugs. And this is a huge problem. At this time point, we have really powerful technologies to try to understand in detail how resistance comes about. And with these technologies, there's many omics platform data, sequence data, and genomics data. And so this huge problem, we have now really nice tools to have a look. But this data alone will not help us to understand this problem. We do need actually new population genetic and evolutionary theory-based ideas and computational methods to analyze the data and to essentially design new experiments that lead to novel approaches in combating resistance. And Alexander's talk was already really nice in terms of very detailed analysis of sequence data. And the sequence data was really nice in the sense that it was basically enumeration of the whole repertoire of these sequences within an individual. So that's really powerful, I think. For this problem of resistance, we need to think about evolutionary theory. And how I think about evolution and evolutionary theory is we have 4 basic levels. And the first level is to discover processes. That would be discovery of processes such as mutation, selection, recombination. I think we are very good at that indeed. We know a lot qualitatively about these processes, but we are still not saturated there. So for instance, in the case of cancer, we discover new mutational processes still. So even though this is a solid land discovery of processes, it's not yet ready. What's even less ready is how we understand these processes. And here, I mean, we need to understand how mutation, selection, recombination together shape the genetic composition of a population. And there we actually need to put numbers. We need to understand whether selection changes the population, genetic composition of the population faster than mutation. So we need to compare rates. We need to measure rates so that we understand these things. And if we indeed claim that we understand something, then of course, in the physics sense, we should be able to make predictions that are useful. So that's, I guess, the ultimate measure of understanding, if you can start making useful predictions. And this is something that gets us to deeper waters in the context of evolution. There isn't much that we can predict at the moment. But there are some very interesting studies popping up. This year, there were some interesting influenza prediction papers out, where the challenge is that the global influenza comes back every year, and the World Health Organization needs to pick a vaccine strain against the next season's influenza. But that selection has to be done 9 months before. So someone needs to bridge that gap. You need to be able to project forward the influenza evolution for 9 months. And people are now making progress in that front by having detailed evolutionary theory-based methods. So there is progress here, but it's still very thin. Then the last level is actually we should not be content if we can predict evolutionary futures, but we in fact want to control evolving populations. And this control problem is actually at the heart of drug resistance. At first, the idea that we want to control evolving populations may sound arrogant, but that's actually what happens in every successful application of chemotherapy, let's say. The clinician is successfully controlling an evolving population. But it doesn't happen always like that. And like I said, the reason for that is that the population becomes resistant to the treatment that it's given to it. And one big reason, let's say in the context of cancer, is the observation that tumors are not genetically homogeneous. So let's have here a look of a schematic view of tumor evolution. So we have first a normal cell here. It has 2 copies of chromosomes. It's a normal diploid cell. And then at some point, it accumulates somatic single nucleotide variant. So it accumulates a mutation. And that mutation then leads to some growth advantage of that cell. And it starts to increase. And on that background, you have new aberrations coming. So what you have here, for instance, there's a gain of a chromosomal segment. Okay. That seeds what we call subclone B here. And this goes further. Further mutations are discovered. And there's even an independent lineage that is living for short duration of time. So what happens now here is that depending how you sample your cancer cell population, first of all, the time point will matter, what you get, and also how carefully you sample the mass. So if you do a naive sample extraction, let's say you would only take cells from here, you would not know about the diversity in the population. And what people can do these days is they can do multi-region sampling of a tumor, or in a blood cancer they can take several time points. So these are kind of ways of trying to get a representative sample of your tumor. And why does this matter actually? Well, if you think about that your population has this much of heterogeneity in it, these subclones actually can be resistant to the treatment. So, for instance, you might have something that works against the blue clone. So you start to eradicate the blue clone with that treatment. But while you are doing so, you actually release space for the red clone to take over the population. That's a process called competitive release. And then you might not have anything against the red clone. And indeed, with clinical data, probability of treatment success is typically higher when the tumor is genetically homogeneous and or early stage. So what I have for today is 2 different lines of research in this context. And the first one is essentially trying to look how heterogeneous populations escape from the application of drugs. And with collaborators, we have here a yeast evolution, experimental evolution assay, which we have exposed to cancer drugs, and we see then what happens. And here, the second part of the first part is essentially looking of some real cancer data, and then depending how we are doing with time, some theory work on how to delay the emergence of drug resistance. So, the part 1A, heterogeneous yeast populations adapting to cancer drugs. This is a collaboration project. We have worked for several years now with Gianni Litti, who is based in Nice, in Institute of Research on Cancer and Aging. And this is Gianni's lab, and the experiments that I'm going to show were propagated by Francisco. So Gianni has developed a very nice system in his lab. He can take 2 strains of Cerevisiae. They have diverged millions of years ago. So one strain comes from North America and the other one from West Africa. And yeast genome is 12 million bases long. And these 2 strains, when they were in the wild, they have accumulated point mutations. And they have more than 50,000 single nucleotide variants separating those two genomes. So it means that roughly every 200 bases there is a SNP between those two populations. And that's a long time scale. Accumulation of mutations. So this is something you can't really do in a lab. But you are banking here on the natural variation. So those mutations have been created in the wild. So Gianni can take these strains and he can make them mate. He can cross them, and he can cross them for several generations. And by doing so, you can create a pool of offspring where individual genomes then consist of segments from both of the parents. So you can see here blue and red coming from these parents. And you can make this kind of a multi-generation cross. It's called advanced intercrossed line. And you break the segments, the 52,000 variable sites, you distribute them to these individuals. And we have actually studied the rate of recombination within this cross. And the numbers are such that after 12th generation of crossing, you have a population of, let's say you have 100 million cells here. Each one of them is going to be different. So it's enormous pool of diversity there. And that's actually very important here, because then the next step is you want to select for something. You expose this population to some pressure, your favorite pressure. In this context, we want to know how cancer drugs affect the population, what kind of variants will help these yeast cells to survive. And you can see then schematically what we expect to happen is that there is some variant that starts to increase in frequency when we do that. And the whole process is then monitored by sequencing the population at some intervals. So this is actually conceptually quite nice factorization of different evolutionary forces. So the first thing is the mutations happened in the wild, that's that part. Then you bring them to a lab, you have recombination. And then the last step, you have selection. And that this kind of factorization will help the analysis of the data. So this last part you can either do sexually or asexually. And if you do it asexually, it means that after this point, there's no more recombination in the population. So, and these two are experimental timescales, while, like I said, this is millions of years. So let's have a look of the selection experiment. So I mentioned that this initial pool, every individual will be different genetically. The genotype will be different based on the rate of recombination that we have. Now we are banking essentially that genetic diversity leads to also phenotypic diversity, because we need to have phenotypic diversity because selection cannot act. If everyone is same in the population, selection cannot act. So selection always needs diversity. And what you see here, we have 5 different settings: fleomycin drug, then rapamycin, hydroxyurea, then some combinations. And in these experimental evolution experiments, you always have control condition. So this control is you propagate it without anything. And let's just have a look of the initial population, whether that genetic variability leads to any phenotypic variability. So what you see here is the initial population. It's done in 2 replicas and some spot assays. So basically seeing how different individuals grow and form colonies under fleomycin. And you can see essentially that yes, there is quite a bit of phenotypic variability in the initial population. Same for rapamycin here. And then if you go and look at the final time point, what you see in the final time point is that if you put them in the control condition, you see that all the pools are growing okay in the control condition after the propagation, but the one that was selected in fleomycin, so the whole duration was under fleomycin, is now enriched with individuals that can grow nicely in the Pleiomycin condition. So clearly selection has done something during the experiment. So these are the time points that we have sequenced, in fact. So 0, 2, 4, 8, 16, and 32 days for this experiment. So we have we have 6 time points. So let's have a look of some of the genetic data. So this is now the control condition, and I'm plotting here the 52,000 segregating sites, and I'm putting all the chromosomes one after each other. So you can see it's about 12 million, as I said. And well, this is now the control condition. And you can see here actually a couple of things. First thing that you notice is that it's not actually at 0.5, this line. Because if you think about the setting, what we would have, the y-axis is counting fraction of one of the parents. So this is now counting the fraction of the West African allele. So when it's at 1, it means that that locus, every individual in the population carries the West African allele. When it's 0, it means that every individual carries the North American allele. Okay, those are the 2 options that you have. It's alleles always are the North American parent or West African parent, and now you are just taking a population measurement with your sequencing. So you're looking what's the population fraction of that allele at that time point under that selection condition. And this condition is here a mock or the control condition. And the first thing you observe, it's not actually centered at 0.5. And this is a manifestation that, in fact, when you do the cross, it's not going to be purely neutral. So even the crossing protocol will itself move allele frequencies. So for instance, there's one region where the North American variant is favored strongly during the cross. So it's actually individuals who have that variant have almost fixed in the population. Okay? So this wiggle is showing you a manifestation of selective forces and processes that went on during the cross before they have seen any other condition. Then the other level of noise here is coming from sequencing. So we have typically 100x coverage. So we are not sequencing infinitely deeply, so we don't get a exact number for the allele frequency in that population, but we have a measurement or noisy measurement from that. And we can model that then differently. And if I filter this data, you see this is our posterior mean for the true allele frequency that you have in the population. And this is now the control condition, and these are the 6 time points for the control condition. And the first approximation.
If you repeat the experiment, you see the same pattern?
You mean the original cross? Closely, but not identically. So if this is now replica 1, there is a second replica 2 of the cross, and you see this is replicated, but there is still some variability which is not, it's more than the noise. So it's not exactly repeated. So there's some stochasticity in the original selection, and but the big picture is repeated, yes. So
how many recombination events do you have here? I mean, just roughly.
Well, let's have a look. This is the population. So essentially, the first approximation, nothing moves in this control experiment. When you look a little bit closer, you see that's not fully true. So there's one region that we haven't seen previously, because previously we didn't go this far in this kind of experiments. But there seems to be some movement here telling us that there is a quantitative trait locus. So one of the alleles, now it would be the North American allele, gives you a growth advantage under the control condition propagation. Because whatever evolution experiment you are making, it's still selective pressure, because you have some protocol how you propagate the system through. But by and large, nothing changes. And now back to Alexander's question. Here actually, this measurement is bulk sequencing. So we take cells from the population, sequence those cells, we lose correlations. So we don't know anymore how the alleles are within one individual. Here we have done isolate sequencing of the T0 population. And this is basically now you take single cells, let them grow, and then sequence those. So you have correlations. You think you are sequencing a clonal population. And what you have here is now these 52,000 loci across 192 individuals and the parental genotypes. So from here you can read how many recombination events there have been. So you can see they are pretty broken down, the parental blocks. Okay. So this is the initial setting. So with control condition, nothing much happens. And we don't have correlations from this measurement, but we have correlations from T0. So that's kind of the setting that we have. Yeah?
These are all haploid cells?
These are all diploid cells. These are haploids. But this is a diploid experiment, what we have done here. Let's look at the condition with cancer drugs. So we have here rapamycin, and I'm now looking at one chromosome in one of the pools, chromosome 8. It has a bit more than 2,000 these segregating sites, and here you can see better the sequencing noise. So this scatter here is sequencing noise, whereas this big line here is selection that happened when the cross was done. Okay? And if I go further to 2 days and 4 days, I'm kind of probably seeing something here. If I filter it with this diffusion filter that we use, I can see that the allele frequencies are starting to shift. And this shift here, it's basically telling us that around this region, there is one or more alleles in these segregating sites that prefer the North American one because it's going down. So this is giving a growth advantage under this rapamycin condition here. And we have worked on how to quantify something like that from the data. And it's a very simple model. We call it driver and passenger model. And essentially what it's saying is that although the recombination was strong, went on for 12 generations, it did not break down all the linkages. So the probability, let's say you have a red allele from one parent here, means that you are a little bit more likely to find a red allele next to you. So there's a correlation there. So this model is saying that we try to explain all that allele frequency movement with one causative allele, driving allele, driver allele, and all the other movement is just because of genetic linkage that was not broken down by the recombination. So they are so-called passenger alleles or hitchhiking alleles. Okay? And here the population size is 100 million or even more, and the time scale for propagation is 100 generations, 200 generations. You can compare those two numbers and notice that the population size is much, much bigger than the number of generations that you propagate. And that tells you that you don't have to worry about genetic drift, because genetic drift will change allele frequencies on the time scale that is proportional to the population size. So we essentially get away with deterministic treatment here. So this is a logistic growth equation. So this is now the allele fraction of the driver allele at locus I. It has some selection coefficient. So it's basically looking at the growth differential between the North American and West African allele. And it then grows logistically like that. And then we can write down how the passenger alleles move. And because we don't have ongoing recombination, their equation of motion is decided by the initial linkage. And that initial linkage to the driving allele we need to parameterize with recombination rate. And the linkage decays. Let's say we have a focal point here, we have a driving locus here, and the further you go from the driving allele, the less linked you are, because there's more room for recombination to break down that linkage the further you go. So that would be this term here. So it's rho times delta ij, which is the distance. And this then leads to allele frequency dynamics, that if you are a driving allele, you do your logistic curve here as a function of time. So this is a family of curves as a function of time. And if you are sitting just next to the driver, you move pretty much identically to the driving allele. And the further you go, the less you move. Okay, so this is the simple model we have used. What we can then do, we can take all that data, that time series data, and we can scan through the whole chromosome, and at each segregating site we can ask, what's the log likelihood that I get to explain this data, given that I'm a driving allele and my neighborhood is moving as passenger alleles, and then we subtract out term what it would be if it's a neutral locus. It doesn't contain a driver allele. And you see both of these things for two different replicas, replica 1 and 2, they peak here where I've shown you the signal. And for the exercise that we are after here, suppose we want to actually find the causative allele here, this is a little bit tricky because you see it's very flat here. And that flatness reflects the fact that if we look at the recombination landscape, which we have tried to infer from a different study, it's showing us that at about 480, it's quite cold in recombination. So the recombination rate is low there. So it means that we don't break down linkage in that region really well. And it means that we can find the region, but we don't have very fine resolution in that region. What is the causal allele? And there is something which we have now validated. There is a CTF8 gene. When it's deleted, it is showing in hybrid West Africa, North America, strong growth under rapamycin. So this is so-called dilution assay. So you have tenfold dilutions of the cells, and you see just how far they grow. So this is semi-quantitative. We are now doing actually phenotyping, quantitative phenotyping of related to this study. But here you can see that in this combination, the CTF8 gives you resistance. It grows then further than in the other combinations. And
Sorry, question.
yeah.
So you see that the recombination rates depend on location.
Yeah.
Did you take that into account into your log likelihood model?
Yeah.
And so my other question is, so the linkage decay, you show a ideal curve.
Yeah.
But can't you just measure it from the first time points? You said that you have the
Yeah, sure. So the question was, just repeating it for the camera, so that did we take into account the recombination landscape? The answer was yes, and it's really simple to take into account because if we go forward here, it just means that this is fixed. Actually, it makes your life easier in the inference. Our earlier version of this problem, we had to learn this as a parameter as well from the data, from the same data as we did the inference. Now it's fixed, it's given that rho, because we have measured that as a part of independent data set. So that's the first bit. And I forgot the second, but the linkage decay. You can do that. It turns out that that's pretty much how you estimate the recombination landscape. But it's a non-trivial problem. It's a problem on its own. So when you have the full haplotype data, there are different methods to get the row, estimate of row from it, but they essentially all use pairwise linkages in some form. That's the step, the logical step there. And well, you can see how it's noisy. It's very noisy. So noisy in the sense it's very rugged. I think that's the better word. So it's very rugged, this recombination landscape, and it makes a difference here in terms of the localization. So let's have a look what happens now if we go forward. We go from 4 days to 8 days. So there isn't much happening from 4 to 8. But then something funny happens at 16 and then also 32. So you can see now the signal is everywhere. It's going all over the place. And here actually things have reversed. They go a different direction. And if I look globally, so this is now the same experiment, and this is a region we were looking at previously, chromosome 8. We look at the whole genome, and you see the signal is all over the place. So this is it's quite interesting because these experiments, when you get the data, you do first a control condition, and you want to see nothing moves there. And you've shown you the data, so nothing really sticks to eye at the global scale. So that's good. Then you move on and check one of the conditions, and you get a plot like this. And you're initially really happy because things move. But of course, if you are looking for QTLs here, you realize that if everything moves, it's not good. So it's a very short-lived euphoria there, like a couple of seconds, and then you realize that actually things didn't work out as we wanted to. But you can actually think what went on here, because you remember these are diploid cells, and they have at each locus then either heterozygous, like one each of each allele, or homozygous in one direction or homozygous in the other direction. And now imagine in that population of cells, this 100 million population of cells, there's one really fit cell. And that really fit cell, in this condition for rapamycin, starts to grow. At some point, it becomes macroscopic. And at some point, it takes over the whole population. And in that limit case, we would actually expect only 3 different levels here. We would expect everything is at 1, 0.5, or 0, because that measurement would only quantify the genotype of that individual. So that's the limit case. You see, it's not quite as clean, but it is conceivable that it's something like that. And we see this kind of emergence of clones in the other pools as well, but we don't see it in every pool. So more than half of the pools we see this, and essentially we have worked out how to do a deconvolution of that signal and try to get the clonal dynamics out of that. I show a little bit more of that in the context of cancer, but we can essentially translate that picture into time evolution of the clones. Yeah?
Why don't you see it arise progressively? And why all of a sudden this clone that takes over as it takes? So it takes over in just a few, in one time point.
Well, you know, first of all, we don't have a uniform sampling in time. So that's one thing. And also, I don't know what to expect, right? I'm just quantifying what's happening in the data. I'm not putting an equation of motion here. I wouldn't know what to write down, right? So we just need to see first what's there. Then maybe at some point we can start to see what would be a good model. Let's say the naive logistic growth, whether that would explain these clones. It's not clear to me. But essentially, the quantification of those clones, there is a computation which is, I think, slightly non-trivial that one has to do to convert that to these clone fractions and their time evolution. So what this is telling us that the rapamycin pool, we identified 3 different clones, and they take, in the end, 85% of the population. at this point, I don't know what's the diversity of the remaining bit, whether this is just that our statistical power is not enough to get small clones, or whether this is still millions of different cells. That's quite an interesting question. I just don't know that yet. And you see some other pools here, and as a part of this inference, we get the genotypes of the clones. So this is now showing you one clone here, the blue clone, and essentially it's telling you what's the genotype in the segregating sites of that clone. And at this point, Johnny with his team went back to these pools and isolated cells so that we would actually have a look if we find clones in these pools. And indeed we find clones, and this is now from that hydroxyurea pool that I showed you the genotype. We have 6 isolates from the pool, and they look actually quite similar, right, these 6? But there are some differences. I make this a little bit easier for you, so I'm not going to count the differences here, but I'm just going to collapse the data. I'm showing you the genotype of those clones. So what you have here, red is now homozygous in one direction, yellow is heterozygous, and blue is homozygous in the other direction. And you see these 6 clones from this hydroxyurea pool. They are more than 90% similar, but you see they have differences. So they have regions where they have lost the heterozygosity. So it looks to us that there's, within this expanding clone that is taking over the population, there's a very active dynamics that is shuffling the chromosomes and is essentially dropping the heterozygous states away. So you see here, these 3 clones all are heterozygous in this chromosome 7 end, but these 2 have lost the heterozygosity. So there has been LOH event there. And why is this important? Because, well, it turns out that from these isolates we can see what mutations are driving, what de novo mutations are driving these clones. So if we have a population that is taken over by one very fit cell, we need to ask what's giving that fitness to that cell? Why is it so good? And one answer could be that it has a very, very lucky combination of this standing variation, the parental alleles, so that would be a viable explanation, or it has acquired a new mutation when we started to select. And this de novo mutation hypothesis is something that we think is correct. And what you see here is a spot assay for these 6 clones, and there is a mutation in RNR2 protein that is changing amino acid, and you can see that without that mutation, cells don't grow nearly as well as with that mutation, whereas in the control condition they all grow nicely. And here is a manifestation why this diversification via the LOH dynamics is so important. It allows actually a very quick mechanism to make the de novo mutation from heterozygous state to homozygous state. So you don't have to discover the mutation again, you just remove the other copy, the wild type copy, and then you copy the segment which has the mutated allele. So in this pool, actually, this R2b pool, we found a different RNR2 mutation, but we found it, first of all, we find it in the same background. So it clusters together. So these are clones that are clearly the same clone, minus the LOH events. But the LOH event has hit the RNR2 locus, and in one of the clones it's homozygous and the other one it's heterozygous. So that's a quick way for you to get a very strong resistance phenotype. Okay, so to conclude this part, we propagated heterogeneous yeast populations under cancer drugs, and we monitored this process by sequencing. And the samples without macroscopic clones some pools all the way, and some pools, the early time points, can be used to identify alleles from the standing variation that give resistance phenotype. And these late time points for several pools show this striking mode of adaptation via the macroscopic clones taking over the population. And they are essentially driven by de novo mutations. And we have developed an algorithm, cloneHD, which can be used to analyze such clones from a bulk sequencing population. And we have identified some mutations driving resistance phenotype, RNR2, RNR4, FKB1 for rapamycin, TSR4-hydroxyurea. And we have evidence for very extensive ongoing diversification using a loss of heterozygosity dynamics within the clones. And we have some evidence that actually hydroxyurea itself is making that rate higher. But that's not as conclusive. So this is acknowledgments for the yeast part. So Gianni's lab, Francisco did the propagation, and then Jonas Varinger lab in Gothenburg, he's doing the phenotyping for these cells, all the isolates that we have sequenced and characterized. And then Ignacio from my group and Andre and Wellcome Trust for funding. And if you have questions for the yeast part, we should have them now.
So this only works only if you have whole genome sequencing, or it could also work if you have exome sequencing for Tumours? cloneHD.
It works for exomes as well. Yeah.
And I was not sure I understood, but when you said that you have a de novo mutation,
Yeah.
could it be also that this mutation existed but in a low fraction, so you could
Yeah.
detect it?
It could.
And then you change the fitness landscape with the drug and then...
Sure. Actually, we don't know that that's not the case, but I think for us it doesn't make a big difference, because if you evaluate do a back of an envelope calculation with mutation rate, the population size, and the genome size, I think it takes about 10 generations to generate any point mutation. So you would probably find it quite quickly. Then the question is whether it survives drift or not. And in some pools, after 32 days, we don't see clones. So that would speak to, at least it's not in high frequency. Like high frequency here would be 1 in 10,000. Then it would be very deterministic when we start doing the selection. So we don't see that. So it's either very small frequency or it's a purely de novo. So that's the.
Can HU induce loss of heterogeneous through break-induced replication, or is it more than
I don't know, but I think people know about that. So it's something. We are not claiming that we have discovered HU is changing that LOH. But that's clearly the mechanism to get very quickly the de novo mutations to homozygous state. So it's.
So you don't expect to have loss of heterodiversity for other stresses?
Well, we see something for rapamycin as well. But you know, what we actually don't have, we don't have from the final population the LOH measurement is actually quite tricky to do for the control condition, because you need to have a clone. And you don't have a clone. So it's, but there are some assays how to do it, but it's not as nice as just sequencing and then counting, you know. It's slightly different.
And technically it's not possible to have even a low coverage single cell sequencing?
That's not, the single cell thing is not a problem for yeast, because you can make isolates, and those isolates you can let form colonies and you sequence that. That's essentially a single cell sequencing. But now if we do that for the control condition, each one of them is going to be completely different. So you can't count the LOH events from there. Okay, so let's move forward to high definition reconstruction of clonal composition in cancer. So here it's again, basically, this is looking at what's happening in a cancer cell population. And just to do that quantitatively requires quite a bit of work, in fact. So I've shown you already this picture. There is a normal cell, somatic evolution takes place, and you accumulate aberrations. Big events, chromosomal aberrations, or then somatic point mutations. And why this matters is because this heterogeneity can be fuel for drug resistance when the clinician is trying to treat the tumor. So essentially, we would want to understand from cancer samples what's there in the sample. So I would want to understand something about subclones within a cancer sample. Let me just define what I mean by subclone. So suppose you have a cell population here, and in that cell population, you look at the ploidy of the cells. You look how many copies of each chromosome the cells have. And you find cells which are all diploid, so they have all 2 copies of each parental chromosome. And then you find a population of cells that have, let's say, gained some chromosome. So now you have chromosome 1 in 3 copies. That's a chromosomal aberration. So you have gained a segment of chromosome. So this would be one way to delineate the pool. So you have now 50-50. You have some normal cells and some fraction of cells with chromosomal gains in them. I can alternatively look at point mutations. So you have cells which don't have any somatic point mutations. And then you have cells which have this mutation here, and you have cells that have this mutation here. So this delineation would give me 3 different cell populations. Or I can look simultaneously at copy number aberrations and somatic point mutations. And in this case, I would have 4 different cell populations. This without the mutation, but copy number aberration, then copy number aberration plus a mutation, and just with point mutation here, and normal cells. In the cancer context, this would mean that you have 3 subclones in your population, and you have contamination from normal. So cancer samples are never 100% of tumor, essentially. There's always some fraction of healthy cells in the population. And it's typically something like 20%, 30%. Can be more. So here it would be 25% is normal, and then the actual cancer part has 3 different subclones. So I would just want to count who is there in the cancer cell population. And it's unfortunately not that trivial, because the sequencing data comes with this kind of a mess. It's short reads reporting from all of these different subpopulations. And it then means that we have to, in fact, reconstruct the subclones from the population probabilistically without knowing a priori who is there. So that's the counting exercise that we have to pull through to understand something from this data. So we have developed an algorithm for it called cloneHD. This is the one we also used for the demixing of the yeast population. It's a slightly different problem, but it's related. And it's a fully probabilistic algorithm that tries to exploit correlations when they exist. Let's say you have longitudinal data, or you have multi-region sequencing, or metastatic samples, or you have correlations across the genome. So copy number change is actually can change megabases of the genome or whole chromosome arm. So even though you have short read sequencing, the actual signal is very strong, and millions of different reads are reporting the same event. So those correlated reads can then be exploited. And this algorithm operates at the level of read depth and so-called B allele counts. I explain you later what this is, and somatic SNP counts. And we can run it through with real data. So with cancer genomics, the data sets and sizes tend to be quite large. So you need to, whatever you develop should be then applicable to real data, so to say. And so when you develop an algorithm like that, you always benchmark with simulations. So I'm just going to show you this benchmark just to explain you what's the data like. So this is simulations. So suppose you have 3 time points sequenced from a tumor progression, T1, T2, T3, and you have always 30% of healthy contamination in your cells. That's the white area. You have 2 clones, red and green, and they then have some progression. So that's some information that we would like to get into. Then these 2 clones have some genotypes. So this is now the copy number state. So it's saying that in red clone has first 2 copies, and the B allele status is telling that these 2 copies are actually both parental chromosomes are present, because you can have 2 copies, but they are the same kind. You might have gained first 1 copy, so then you have 2, 1, and then you have lost the single copy. And that would be a big aberration. That would be LOH, loss of heterozygosity, copy neutral loss of heterozygosity in the cancer case. And here you have some gains, and this is the green clone. Then you have the same thing. You have some gains, you have losses. Here you have copy number 1, you have lost a copy. And here you would have the somatic single nucleotide variants, and what's the genotype. Genotype here means how many copies of that mutation you have in your genome. So this is the hidden data. This is what we actually are after. Then we get, when we sequence a pool of cancer cells, we would get read depth track. It's looking how many reads hit any given region in the genome. And that is basically directly telling you something about the number of chromosomes in that location. So this is now emission, essentially. This signal is emitted from the hidden state, which is the mixture of these clones with these proportions. And I have just to guide your eye, the real breakpoints are shown in this plot. But you can see some of them are very clear even without these lines. And this P allele track is a little bit more noisy, but you can see that there is so-called allelic imbalance. It should be at 0.5. For healthy individual, this is basically just plotting the germline heterozygous loci, and they should emit at 0.5. So it should be always at that allele fraction should be at 0.5. When you have imbalance there, it's you see, for instance, there's some imbalance here. So the points are ever so slightly not consistent via emissions from 0.5. And then the noisiest track of all is the somatic SNV fraction, because that track is not correlated in the same sense as the copy number. So this is quite easy to see by eye what's going on, because these are big events and lots of reads are reporting them. So this is the input data, and then the output data is going to be the clone fractions and a posterior probability for the clone genotype. And for instance, this example with 3 samples, we deconvolute the fractions right. There's one region which we misassign, but it's pretty much spot on here in terms of the input data that goes in. And we have then quantified how well this kind of thing is doing. And it's very important to, first of all, use simultaneously all these 3 data things. There are many degeneracies in this problem. So you can have several solutions explaining equally well the counts. And one way to get rid of that degeneracy is to use these 3 tracks simultaneously in consistent way. And the second thing is if you have multiple samples. And here the key is that the multiple samples, if you're lucky, when there's tumor progression, they have the different subclones in different fractions. And that's very powerful in killing the degenerate solutions, because you see the same system from a different angle. And that's really helpful. So I'm just going to show you some real data. So we have done a case study using a CLL, chronic lymphocytic leukemia patients, sequenced at 5 time points. I want to thank Anna Shu, Jennifer Peck, and JP Katsia, who helped us with the data access. And here's one patient. So what I'm plotting here, 5 time points, And this is now somatic single nucleotide variant fractions. And you can kind of by eye see already that there's something interesting happening here. And this is the retep track with some tiling for 5 time points. And you can see there's something interesting in chromosome 8, right? And you can also see the gender of the patient if you look at X chromosome. So that's a male patient because it drops to 1/2 the amount of reads reporting that. And this is then the input data we take. And now we want to understand what kind of clones are in that population. And this is our interpretation. So there are 3 clones. This white area is normal cells. And the red clone declines, then the blue comes and takes over the population. The green clone hovers in the population with some frequency. And again, we are not trying to make any statement about the evolutionary dynamics here. We're just seeing what happens in these samples. But, you know, I think it's not far-fetched to think that one could fit a model here. So this doesn't look erratic at all. So definitely there's progression. Something is going down, something is taking it over. We need more data. I think with one sample, one cancer, it might be pushing it too far. It's better to have maybe tens or hundreds of these kind of evolutions. And then you could try to parameterize them. And in this study that reported this data, they did what's called targeted deep sequencing. So they went after 20 or so coding mutations and did 100,000x targeted deep sequencing of them. And from that data, they deduced evolution of this kind. And this is their healthy. It's very close to what we have from the whole genome data. There's one small clone we don't pick, which is only present in time point A. But essentially, with this whole genome data, we recapitulate what they have doing the targeted deep sequencing. So this is kind of interesting because this is for me, this is getting to see what's there in the cancer cell population. But basically, very keen to get this kind of tools and insights to bear some clinical help in clinical practice. And for that, well, we want to actually produce assignment of mutations to these clones. And you can see here what we have done here. So we can say actually what single nucleotide variants belong to each clone. And also, so not only the progression of the clones is interesting, but their genotypes are interesting. So here we can see that the blue clone actually has this aberration where it's loss of arm and gain of arm on chromosome 8. But there's still, suppose you want to use something like this in the clinic, there's still a fundamental flaw here. This is really nice, high resolution information, but it's retrospective. So that's really it's a problem. So it's good for learning about tumor evolution, but not so good to actually see if it would help in the clinical setting. And we wanted to have a look if there is some room for that. So how well would this work? And we basically formed 5 different data sets. So we divided the whole data set to 5 different ones. So first you have one time point only, then the first 2, first 3, first 4, first 5. And then we try to essentially repeat the inference that we have done. And let's see how well we do. So these time point switches are corresponding often to change in regime. So you can see also that fitting a fitness model here might be quite challenging because the fitness landscape is changing. So you probably need a little bit denser sampling, keep something constant, and even sample while you are keeping the treatment the same. So if we do that partial inference, what I have here, this is with all data. So the first time point, we get the fractions nicely. Second time point, well, it still looks pretty much the same. It's a little bit different. But this is now the critical time point here. So essentially, here we see that the healthy tissue has gained in frequency. So we think we are doing well. But now the key is, do we see actually what the subclones are doing? And we see it pretty much here. So what we see here, that although things superficially are working because the healthy fraction is increasing, there is a new clone that is taking over the population, the blue one. So the treatment is clearly working against the red one. The blue one is now taking over the population, and then indeed there's nothing against that. Yeah? Sorry? Let's have a look. So it's after, C is after 6 cycles of B. So this is telling us that this kind of data could be informative already when you are trying to do it near real time. So whatever is your turnaround time for your sequencing, so it could work quite nicely. Let's have a look how we identify the clones. So these are only the clone fractions. So if you look at the posteriors here, first of all, we see that the red clone, unsurprisingly, we only need to see the first time point and we nail it. It doesn't change anymore. Whereas the blue clone, we actually only start fixing after time C. And in fact, I'm not sure if I have the clustering plot here, but this blue clone has nothing to do with this one. So it is something different here. So, but from here onwards, it becomes what we identify with the whole data set. Oh, here is the clustering plot. So this blue clone here clusters together with the green one, and it's only these we see the real blue clone we see first time only with 3 data points. But at that point, we would basically be in a position to say that the treatment works against the red clone, but there's possibly a seed of resistance in the sense that there's a new dominant clone in the population, so that the blue one is taking over. So that's possibly valuable information. We have analyzed some other samples with this, actually quite a few so far. This is single time point sample, so it can be analyzed with this kind of methods. So this was published by the Sanger Cancer Genome Project 2 years ago. It's a very high quality breast cancer sample. It was sequenced to 188x, so it's very high coverage, unusual. They wanted to study very carefully that, and they did a really beautiful paper out of that. It pretty much reads like a detective story, so I would read that one, the life history of 21 breast cancers. And our ambition with this kind of an algorithm is to essentially do pretty much automatically that analysis, because it was time consuming for them and they used lots of resources and time for it. And we pretty much get automatically the key results of that. So that's quite nice. But things work much better for multi-sample data sets. So we have developed an algorithm which can be used to reconstruct subclone fractions, their copy number, and SNV genotypes from whole genome and GS data, exploits correlations, and this is an interesting conceptual point. So in cancer genomics, let's say a breast cancer can have 20,000 mutations, and maybe 10 of them are of some importance in the sense that they are so-called driver mutations that give you the phenotype. So it's a very difficult problem, and it's something that many people are working on, where they are trying to discover these driver mutations, these 10 important events out of these 20,000. However, what we have here is a different problem, because we want to see who is there in the population. We are better off with many mutations, because we actually use all these passenger mutations and their movement, their joint movement, to deduce what kind of cells are in the population. So in fact, for this problem, the passenger mutations become an asset. So that's an interesting change of fortunes there. And this reanalysis of CLL whole genome data recapitulated clonal evolution, that the people who generated the data found out by targeted deep sequencing. And I mentioned this already, once we have a large number of such evolutions, then I think it's the right time to start to parameterize those models and understand what kind of fitness landscape is consistent with that evolution. But we need a little bit more data. And this is, I think, quite promising, that this kind of mimic of a real-time monitoring seems feasible. And also this kind of subclone-specific computational analysis is interesting future direction. And this is work was done within my group and is spearheaded by Andre. He's a very strong quantitative person, and we have benefited with Gianni's group and interactions. You saw we used this kind of approach to the yeast data as well. And then people from Sanger Cancer Genome Project for discussions, and also several other people for discussions. And Chris Illingworth, my former postdoc, he is group leader now in Cambridge. He was also part of the work. And Wellcome Trust for funding. So I think I have a third part, but I think basically, why don't we take questions from here, and I think this is good time to wrap up as well.
I have a question about the clustering process. The old clustering of one of the sample samples, does it remain the same if you change the clustering algorithm?
Question was, what was the clustering that I have showed? Yeah, and how robust is that?
Yeah.
You know, this is completely standard hierarchical clustering. It's not a phylogenetic tree or anything. It's basically just looking at the clone identities. And I'm sure you could use any clustering for that. So it's not anything deep there. Okay.
This is very impressive, really. My question is, how much could you get without doing full inference, just by doing some sort of summary statistics? Because I imagine you can get the fractions of the clones.
I'm not so sure. It's at least with the cancer, it's really noisy. So you need to exploit the correlations. So the first thing, you can basically, let's say for the yeast problem, you don't factor in the persistence coming from the finite recombination rate. And you get, okay, I'm probably seeing what you're saying. You will get the fractions right, at least the total fraction. Yeah, because that you can kind of already see by eye. But you get the genotypes completely messed. Yeah. And also, I think you don't get the sub-fractions right, but the total fraction is easy to get. Okay. That we could just basically deduce by looking at the heights from the filtered allele frequency curve. You're right. That's pretty much how we started. Okay. So.
Because I think the thing you can't get is when here you essentially see in time point C that the blue one is going to take over and be the next spread, which should influence your next treatment.
Yeah.
That kind of thing I don't, and specifically the identity of the blue clone.
Yeah.
You can't get.
Yeah. I think with the cancer data, it's more tricky. I think with the yeast data, because it's basically diploid, you can get the fraction right by reasoning if you have done the pre-filtering, so that you have a line that you can see. If you have only the dots, you can't do even that. But for the cancer data where there are ploidy changes, and this CLL sample was very nice, it was almost diploid. It's very tricky, so you really can't do it in your head, I think. So that's, but then you shouldn't, because you can just, you can do it properly, essentially. That's what I'm saying. Yeah, but that's kind of how we started with the yeast problem. You can reason and you get the fractions, you get an expectation what you should find, and then you go forward from there. there's one thing I wanted to show you before we finish. Namely, we had this summer we had a 9-week program on drug resistance, and it's obviously gone, but all the talks are, most of them are online. I think pretty much most of them are online with slides as well on the topic of drug resistance. So I think that's something you might want to have a look if you're interested on these things. And the part that I will skip now, I think I can't go through in this kind of time, is about doing some theory how to avoid the emergence of resistance, or at least delay it. And we have been working on that using stochastic control theory, and there's a manuscript in the archive, so you can have a look it from there. Okay, so thank you.
This is sort of a new question, but in the title it says you also want to combat the evolution of
Yeah.
drug resistance. So with your program, you can sort of see what kind of
Yeah.
different clones.
Yeah.
But then you see some that like are resistant to like everything. And I don't know, is there something you can do there?
Yeah. So I should have moved on quicker, because I think you remember my line of things: discover, understand, predict. So I essentially talked today only about the understanding bit. The control theory part, I think, is kind of what can we do to change the situation. That's the third part of the talk. And so there are quite powerful ideas around how you should go about it. And well, we can talk after.
If you want to take 5 minutes, because you're definitely on time, so if you want to...
I can say 5 minutes from that, but you know, I do want to keep it in 5 minutes. So that's okay. So maybe it's just me who is tired of talking. So yeah, I think I will show you Gatenby, because that's really a powerful thing. So how to delay the emergence of resistance. So there's a very nice paper that I read a couple of years ago by Gatenby et al. And essentially, Bob Gatenby's idea was that perhaps all-out chemotherapy is not a good idea, because it doesn't seem to work. You have this competitive release, and then you have resistant clones. Against the resistant clones, you have nothing left. This is exactly what the question was about. And he did some experiments. So his idea was that cancer as a dynamical, or Gatenby et al. So their idea was that cancer as a dynamical process, the protocol that you use to control it should also be dynamical. And he said even further that we don't necessarily need to eradicate the tumor. We should be talking about maintaining it at some level. And what he did with an ovarian mouse model, ovarian cancer mouse model, he had 3 different protocols. One was doing nothing, then standard regime, standard chemotherapy, and then what he calls adaptive regime. And the adaptive regime is essentially when the tumor increases in size, you increase the dosage. When it's shrinking, you decrease the dosage. And your goal is to maintain some tumor population size. You are not even trying to kill it. And that's the green line. So you see they can do really well. And this is a different replica experiment, different mice. And so Bob is telling that they can these days maintain these mice indefinitely. So that's quite interesting. And also, the actual dosage that they need to have this balance is decreasing as a function of time. So these are quite interesting observations. And this paper I thought was brilliant when I found it. And so we have basically thought about how to formalize this intuition. And if there's a systematic, optimal way to control tumor evolution, and how feasible is the optimal control, and what kind of minimal models of resistance resistance to treatment in cancer one should be studying about. Yeah?
Did we sequence the mice with the green line? Did the tumors stay at the same fraction like they did with the clones?
Not in that paper. There's no genetic data in that paper.
What is the idea? That was the idea to keep the
Yeah, so
mutation fractions the same.
yeah, you would. I think it'd be very good to essentially good to have this line now reporting some genetic data which would tell something about cell populations. That'd be very, very important, and it'd be very important to see how it is here. So presumably these are all resistant cells, right? But here there must be some balance because you can still maintain it. But yeah, that data is not yet available. So anyway, so this is basically, these kind of questions are then interesting if you think this adaptive therapy idea is good, and I think it's a strong idea. Another strong idea is combination therapy, obviously, but also with combination therapies you can think of the time-dependent component. And so we have basically looked into stochastic optimal control as one way to analyze such systems, and it's actually a very mature field of study. So engineering and finance, economics, it hasn't been used so much in biology, but some cases, let's say, I think immune reaction studies, people use this kind of control theory methods. I at least recall some stuff from there. And so what we have done here is we have basically looked at the easiest case, which is still non-trivial, and that's the Wright-Fisher evolution problem. So Wright-Fisher evolution, maybe you already were introduced to that during this course, but you can have a one locus, two allele version of it, which is very simple. So you have two alleles, A and B in the population. They can mutate to each other with some rate. They have some growth rates, and the difference in the growth rate then relates to selection, which one is fit or not. And this X is counting the fraction of given allele in the total population of N. And you can analyze such a model with the Fokker-Planck equation, and you can write the probability density of time development of the probability density of observing some fraction of allele A in the population after time t, if there's some selection in the population mutation. And this last term is here, genetic drift. So this is noise in reproduction. So you see, this is not nothing to do with cancer yet. For us, this was a kind of way to get into this stochastic control. And we asked here a simple question. You have a population of Wright-Fisher evolution, and you start with some frequency. If you do nothing, if there's selection, the population is going to fix on a time scale which is n generations divided by the strength of selection, proportional to that. So that would be the uncontrolled system. And we asked then, suppose we can change the balance of selection between the alleles. So that's our control measure. We can change that balance. And what should we do if we want to maintain the population polymorphic for as long as possible? So we care for diversity for this problem. And well, so selection goes to selection plus this control parameter. And we keep it simple so we don't pay any cost for applying the control. And we then want to keep the population polymorphic. And what happens here is that you can see that your control parameter is entering the equation of motion linearly. And if there's no cost to apply that control, it means that you never use any intermediate values for your selection parameter, your control parameter. You either don't use it or you use it at its maximum value. This is called so-called bang-bang control, all or nothing. And then the question for this problem is, where should I do that switch between these two values? And these are typically really hard problems to solve analytically. For this particular problem, we can guess the solution, and then we can verify that with numerical solution of the value function iteration here. So here it means that there is a threshold that the mean survival time of the population will peak if you switch at this point, if your control is -30 and your selection is 10. So you go a little bit to that boundary because your control is stronger. So here you have effective selection of -20 and here you have 10. So this is something, and if you do that, you can actually maintain the population, not indefinitely, because still a stochastic system. So at some point you're still lost. You are unlucky against the fluctuations. But I think we can maintain it to 8,000 times the 8,000 times n, whereas without any control, it's going to be some fractions of n. So it's going to stay alive for a long time if you do that. Well, this is very unrealistic in the sense that to do that calculation, we would need to have instantaneous information without any interruptions and accurate measurements. And that's in a real life setting is never, at least for biology, never the case. You just think about your sequencing budget. You can't sequence every time point or every generation. As simple as that. So you immediately have a sparse sampling in time. And these are interesting problems because that changes the calculation. So if we now try to control the same population, but if our measurements are arriving at some intervals delta, and we don't take that into account, we start amplifying fluctuations and we start overshooting. Because here, for instance, we start from here and we say we need to apply selection, but we apply it the whole time until the next measurement comes, but we are almost killed to the other boundary here. So this kind of a naive control with sparse monitoring leads to overshooting and amplifies fluctuations. And you can try to do something better by anticipating that dynamics. And here again, to get an exact solution would be very difficult, but we can look for a good performing protocol from a small space of protocols, which is basically our ansatz for this is you apply selection for some unknown time which you need to learn, and then you neutralize the system because you want to stay somewhere in the middle before you get the next measurement. And if we do something like that, we see that we do quite well. And here is an axis which is telling you when the next data point is going to arrive. And if it's, in this case, we have perfect information, it's here, so you can be closer to the boundary because you know the next data point is coming quickly, so you can take more risk. But if you actually know that the next data point comes very late, so you are basically just steering to the middle. And we characterize these two things as playing to win and here playing not to lose. So these are the behaviors. And if we look actually how well we then do in terms of keeping the population polymorphic, perfect information, it was something like 8,000 generations. And then we do naive protocol, which is we just apply the perfect information scenario. It's still a good thing. It's much better than doing nothing. But this kind of preemptive protocol is outperforming it quite significantly. So for instance, here it's keeping things 8 times longer alive. And 8 is a big number here because if you think the cancer problem and you want to maintain sizable, at some acceptable level of tumor burden, let's say, eightfold, making that interval 8 times longer is really helpful, because what you in fact want to have, you want the person to die for something else, not for the cancer. You don't have to get rid of the cancer, you just need to make that irrelevant, right? So that's the logic. So these kind of gains could be quite nice. And we have then played this kind of calculation through in a minimal model for cancer evolution, and there are some interesting things that come out from it. So here is something like metastasis-free fraction. That's how we quantify how well we are doing. And here you see if you do nothing, it's the blue line. You basically, after some characteristic time of 10 units, you always are metastatic. And the all-out chemotherapy is not doing much better. There is a metronome therapy. It's something that you use time-dependent protocol, but you don't derive feedback from the system. So you just decide it from the outset, this is what I will do. And what we have done here to compare is we have optimized the best values for those switching, and that therapy is doing quite well for this simple system. But the adaptive therapy, which is deriving information from the actual population, is still outperforming it, and it's doing much better. And essentially, if there's one thing I want to get across with this last very quick 5 minutes in the control theory, is that it's a very interesting field, and I think it's going to be worth to be exploited. And we have worked now one problem there in the context of cancer, and that's there available for reading. Thanks.
Are there more questions?
So, what if you add cost of applying the chemotherapy? Have you looked at that?
Yeah, it's a very good question. So we first thought that we don't apply any cost, and our reasoning was that we don't want to make a conversion to money, because money is a big thing there.
Thinking of cost, this point of you're going to die.
Yeah. That's absolutely, that's a great question. So we actually, we now in a new version of the manuscript, we have factored that in, because reviewer gave us that very helpful comment. So you can think about it as an additional death rate, and then it's in the same scale as the metastasis, because the metastasis or next catastrophic event for us is something that after that you have lost. But in the same sense, when you apply control, you apply the chemotherapy or targeted therapy, it has a cost, and it has also some death rate associated to it. And then those two things are exactly the same thing. And it does change things interestingly. So it really adds one, so these problems are very rich. So if you now take the cost into account, it alters things, but we have done very little there. We've just done this one extension.
Have you tried to include an adaptive delta? So, by which I mean, for example, if you know the fraction is at 98%, you don't want to put very negative selection.
No.
But then you also want to sequence very
Yeah. I like that. We haven't done that. Yeah, we have discussed what should you. Yeah, that's a really good. Yeah, absolutely. I think we should keep in mind that we ultimately are empowered to change the delta. So, and we should probably think differently about it. Now this is very passive, right? It's really, that's a good point. But we haven't done it, so we don't know. For the cancer part, we actually were talking more about trying to do the problem without getting the inner composition of the tumor. So without getting the cells, the cell fractions that are resistant versus the cell fractions that are susceptible. So suppose in the KDNB scenario, they only follow the total population size. And now the point is that you actually would want to get the inner composition because you would do much better than in the control. So that's kind of the limited monitoring versus high resolution monitoring that we have done for cancer. And this for the Wright-Fisher evolution, it was the time aspect we have looked into detail.